Can you hear me on this?
Yeah.
Is this working? Good?
Speak, like just
ABCs,
ABCs,
ABCs, good? All right. Yes. How long, what? I got here at about 7:40 this morning. Technically, I could leave right after this. I'll probably stay and prep a little bit for my organic students tomorrow, but probably right after this. Huh? Why? I suppose. I mean, this morning sessions were small, like everybody fit over there. All right, it is officially 3:30. We're gonna get started. I'm a pretty loud person. I'm not gonna put the microphone on unless it's requested. Can everybody hear me okay? Yes, this one is for the recording. If at any point you can't hear me, just let me know and I can put the other mic on. I'm usually told I shouldn't because people get mad at me. I'm gonna go through how today's gonna work. By raise of hand, who is in Dr. Kutsuli's class? Short lady with the fluffy hair, cool. Who's in Dr. Kopashkikov's class? Okay, got a decent number of you guys. I am the SI leader for Dr. Kutsuli's class. However, when I took biochemistry a little while ago, I took it with Dr. Kopashkikov, so I have an idea of how both classes are set up. Allegedly, this is a 4,000 level biochemistry class, and so I'm hoping that everyone has come prepared since our finals are coming up so quickly. So what I will request is that as we go, I do have material prepared for you guys. However, I would like, if you have questions, to please throw them out there. My answer will be one of two things. It'll either be I know and this is what it is, or it'll be I don't know and I will tell you that, okay? So feel free to ask whatever. The worst thing that can happen is that I don't know it and I'll tell you that. Are we all good with that? Does anybody have any questions? Yes? No? I know there was a tad bit of confusion about how this would be structured today. But I did do a practice test that I posted for you guys. So if you go to UCF SARC, well, if you Google this, SI Google site, and then you can type in UCF for safe measures, you'll get our page. Come here, go to chemistry, and then biochemistry with Adam. It is currently December 4th at 3:30 p.m. We are all in the student union. If you scroll down, there is a final review section. There are 3 documents here. The first one you should look at is the Word document. When you guys open that up, you're going to get a short little summary. Here are some links. These are to the old reviews that have been done. One is by myself. One is from Amanda C. For those of you in Dr. Kopashkikov's class, I hope that you guys know her. For you guys in Dr. Kutsuli's class, you can go see her tomorrow because she has a review at who knows what time. 12:30, yes. You guys can go see Amanda at 12:30. This first link is me from last year, and we're going to stop this from playing because, good Lord, I don't want to listen to my own voice. Whoa, there we go. If you guys put this on, though, this will be the multiple choice practice exam that I had made last year. It's posted for you guys, so it's there. My suggestion is go and try that, attempt it, and then go listen to this and work out the solutions. If there's anything that looks like a typo in there, I corrected it in that video and been like, oh, this was a typo. That happens. When I write 40+ multiple choice, typos occur. What else? Next link down is Amanda's old review. If you click on that, the exact same thing will happen. You'll be taken to a YouTube page, and you'll get Amanda C's review for fall. Good. Amanda's wonderful. She actually SI'd for this class when I took it a while ago. So go see her. She knows her stuff. And then the other one is someone named Jessica. She was an SI leader that I don't know. I've never met her, so I cannot vouch either way if she's good. I can vouch for Amanda. Amanda is wonderful. All right. I have never met Jessica, though, so I have no idea who that is. And that's it. I'll go through that in a moment. For those of you who have never seen this before, I have a website. It is wonderful. Well, that's what I tell myself, at least. If you guys go there, you'll pop up a thing, and it's got all sorts of chemistry stuff, but if you go to Biochemistry I, topics from biochemistry. The beautiful thing about this is that it's listed by functional group or by the type of macromolecule or the topic, so it does not matter whose class you're in. Within each page, there is a relatively large number of topics. For instance, if you go to signaling pathways, there are 2 PowerPoint overviews of the 2 of them. The one I did not have time to make a PowerPoint for, I apologize, but 2 of them have those, and we're going to go through those in a minute. There are some handouts for the RAS and the heterotrymic G proteins that you can fill out. If you go to, say, amino acids, that one's a little more thorough. It has some handouts in there about amino acid memorization, some links to websites that do a better job of explaining than I do, and then some other stuff. For instance, on hemoglobin and myoglobin, there's a worksheet, there's an explanation, there is a review of the concepts. Good, and there's stuff for muscles and antibodies as well. But if you go through each tab, there's a bunch of stuff in there. Feel free to look through them. Answers are not included, so if you have a question, you have to ask Amanda tomorrow or ask me today, all right? We will start with the signaling pathways. I want to talk through these real quick, just because these tend to get people a little crazy. There is a lot to know. For Dr. Kapashnikov's class, if I remember correctly, you guys should know about the same level of content as Dr. Katsoulis. For Dr. Katsoulis, you guys have to be able to make predictions about the pathway, what's going to happen if you do X, Y, and Z. How will concentrations be altered given a certain condition? So an overview, the one we'll talk about first is the receptor tyrosine kinases, the RTK pathway. This involves phosphorylation of tyrosine residues within certain proteins. It triggers a set of intracellular events, and most of this is controlled by RAS, which is a membrane-bound protein. And then there are what are called GAPs that help speed that process up. The first process that we care about is signal across the membrane. A receptor comes in and binds to these. Now, these are kind of unique in that usually they are subunits floating around within the membrane, and when that when that receptor comes, they dimerize. The exception to this, in this chapter, you guys learn about insulin. Insulin happens to already be dimerized, and so when that thing binds to it, all it does is it causes those subunits to get closer together in the cytoplasm, and they autophosphorylate each other. Depending on the receptor, they can have anywhere from 1 to 3. The example on the screen shows 3. However, there are some proteins that only have 2 phosphate groups on each domain, and so it'll only bind to 1. And this just is protein specific, but that's not very important for our purposes. The structure of these guys, it has 2 beta domains down in the cytoplasm. They pass through the membrane. They're a transmembrane protein, and they are linked via disulfide bonds to some alpha subunits, which are also linked to each other by a disulfide bond. And this one is insulin specifically, okay? For non-insulin receptors, it does not necessarily have to be in a dimer already. They may not be. They may be individual subunits that dimerize upon binding. Okay. Note that the alpha subunit is the part that's going to bind the ligand, the thing that's outside of the cell that's telling it what to do. And the beta domains are going to be the ones that come together and phosphorylate each other. Cool? All right. As the ligand binds the receptor, the cytoplasmic beta domains autophosphorylate each other. So as they get physically closer together in space, some chemistry happens and they phosphorylate each other. Insulin is one that has 2 phosphate groups on each beta domain. In this picture, this was my sad attempt at using paint. Note that those beta domains should be a little physically closer. The picture in your textbook does a better job, but they should be physically closer together in space. Everybody good with that? Good. So I have some questions for you. The insulin receptor shown in the previous slide was isolated from a sample treated with BME, beta-mercaptoethanol, and run on a gel electrophoresis. How many bands are expected and why? And our second question says, assuming the beta subunit is made of 306 residues, what would its approximate location be on the gel, assuming units of kilodaltons? As a note, these PowerPoints I posted these on my personal website under signaling pathways for you guys. The answers are not included, so you guys want to make note of these, okay? So for the first one, what does BME do? What is that stuff? Beautiful. It's going to cleave those. These are reducing agents. Go through and cut those disulfide bonds, And if we remember correctly from that structure, those two beta subdomains, they're each individual proteins, and the two alpha are individual proteins. So I expect a total of 4 fragments. Sounds reminiscent of IgG, does it not? However, since the alpha domains and the beta domains for this insulin are the same, how many bands do we expect on our gel electrophoresis? Just 2, right? Because those migrate based on size and charge, and in this case, they're the same, they're going to overlap. Everybody good with that? And then what we're saying is, assuming the beta subunit is made of 306 residues, what would its approximate location be on the gel? Now, if I remember correctly, Dr. requires you guys to know the average weight of amino acids. Yes, hopefully we know this by now. About 110 Daltons is the weight of one amino acid averaged. And if we're saying it's 306 residues, what we can do is we can multiply that by 110 and say that that's going to be the weight in Daltons. Since it's asking for kilodaltons, and that's typically the unit on these gels, we're going to divide that by 1,000. Good? And so what we get for the first one is that we expect 2 bands because there's 4 parts, but they're identical. All right, remember the yellow lines are representative of those disulfide bonds. Those are going to get cleaved. And then we're going to say 306 times 110. Since we have that averaged weight, we're going to divide by 1,000. And so we expect the beta subunit to show up around 33.7 kilodaltons. Good? Any questions on this? Yes? If you're in Dr. Gutsuli's class, you know that she likes these sort of questions, right? So we're going to be able to do this on the final in case it shows up. I think she gave it to you guys on the second exam, if I remember correctly. All right. Quantification of receptor ligand binding. We can do some math here. This is very similar to hemoglobin and to enzyme kinetics in that how these things bind do so in a predictable manner. We have a graph up there that shows concentration of ligand versus percent binding of 2 different ligands. Which one has a higher affinity for the ligand, the red or the green receptor? At a lower concentration, which one binds more? The red. So which one has a higher affinity? The red. Everybody good with that? This can be expressed mathematically in a few ways. The first equation says that we have a receptor and a ligand, and they come together to form a complex. It's denoted as RL. We can also make a note and say that the total receptor is equal to unbound plus bound receptor. Is everybody good with that? All right. We can from there write a dissociation constant. Don't forget that in most chemistry classes, we write these things saying that equilibrium is products over reactants. For a reason that I don't quite understand, but I just know that they do, biochemists like to write things as reactants over products, and they call it a dissociation constant. That shows up a couple of times. Are we all good with that? Yes, hopefully. That can be rearranged using that equation from total receptor to the third equation written on the screen, and that can be further rearranged to say receptor bound divided by receptor total, which can be expressed as a percent, all right, and that makes math nice and easy, and that's equal to ligand divided by the dissociation constant plus the ligand concentration. Okay? And that particular equation is useful because what we can do is if we know the percent bound or the percent needed to be bound, the dissociation constant, we can calculate the amount of ligand that's needed. Is everybody good with that? And these 2 equations can be derived if you know this and you know that. All right, you don't technically need to memorize all of this if you know how to write a dissociation constant and you know how to do algebra. Okay, so I did not put the derivations up here, but you could do them if you needed to. All right. So the question says, in the graph below, what concentration of ligand is needed for 30% of lower affinity protein to be bound to the ligand? So which ones are lower affinity, red or green? Green. And so what we're going to do is we're just going to read across at 30% bound and read our way down. And I'm eyeballing that. Looks like it's going to be about 10 nanomolar. Yeah, right about there. 9, 10, 11. Those are all reasonable. Affinity. Yep. And then note, we can tell lower affinity because it requires a higher concentration of the particular thing that it's binding to in order to reach the same percent bound. Does that make sense? Good. All right. A cell has 115 receptors for a certain ligand. If only 20% of the proteins must be bound to achieve maximal response, what must be the minimum ligand concentration? What must the minimum ligand concentration be to achieve maximal response? Assume the dissociation constant is 600 picomolar. There are one of two ways to go about this. If you know the equations from the previous slides, you could just plug and chug here. If not, you'd have to do some algebra and figure it out again. Good. And if we do that, which I already have, we can say that R bound divided by R total is equal to L divided by K plus L. And again, that's an equation that was shown on the previous slide. We know that the percent is going to be 0.2. Don't plug in 20 there, okay? 20 is not the number we use. Whenever we use percents, we always plug them in as a decimal, all right? Never, ever plug in 20%. I don't think I've ever seen an equation that actually does that. We're looking for the amount of ligand. We know the association constant, which is approximately 600 picomolar. Since this is all nice add and divide and units cancel, we can leave it in picomolar. For the most part, you should convert to molar, okay? For Dr. Kapashnikov's class, he is notorious for putting things in really nasty units like nanomolar and femtomolar. We got this on the first exam. Yes, everybody remembers this, right? And you had to convert everything to molar and plug it into the delta G equation. I hope we remember. I saw your guys' exam, and that was on there. But it's likely that'll show up again on your final, okay? So be able to convert between any of the units that he told you. And when we go through and simplify that, we end up getting that the concentration should be approximately 150 picomolar. Good? Any questions on that? No? All right, kinase cascade. Certain receptor tyrosine kinases directly phosphorylate their target, but many do not, and instead go through this signaling cascade. The main receptor of the insulin RTK, they're known as insulin receptor substrates 1 and 2, denoted as IRS1 and IRS2. Sounds like the people I have to see every tax season. The insulin receptor interacts with the domain of IRS known as the SARC homology. That SCR is pronounced SARC, like the place that I work at. And that's denoted SH2. These domains bind phosphorylated tyrosines with much higher affinity than the corresponding serine and threonine residues. And this is important because this allows for some selectivity there. So the tyrosine ones are going to bind much more with a higher affinity and hence more selective than the serine and threonine. This selectivity is due to size restriction. So within that, let me get up here so I can show everybody. Within binding pocket, so within that binding pocket, there is an arginine residue that's fairly deep within there. And the tyrosine residue, everybody has these memorized, yes? I'm hoping so. All right, if you don't tell me. It'll just make me sad. But when we take that phosphorylated residue, as it goes into the binding cleft, the serine and threonine are significantly smaller, right? About approximately 1 phenyl ring smaller. And so they don't quite fit inside, and so the binding doesn't occur as well. Whereas tyrosine fits perfectly inside that receptor pocket and will interact with that arginine residue. And that allows it to bind tightly and promote a response. Does that make sense to everybody? Yes? And the residue that's in there is arginine that interacts with the phosphate group. Cool? All right. Binding of a receptor by autophosphorylation in the beta domains results in the bound phosphate binding to an arginine residue in the SH2 domain of a GRB2 or SEM5 protein. And as that occurs, it simultaneously binds to a proline-rich SOS residue. Okay? So as that SH2 domain of the protein binds on there, a SOS domain attaches as well to the SH3 portion of that. And that whole portion allows the SOS to interact with RAS. Okay? The GRB2 is the protein found in mammals, and SEM5 is the one found in nematode worms. Your book makes a fuss about that. I'm not sure why. My best guess would be that the nematode worm was the one that was studied initially, and that's the one they know a lot about, and they have applied that to the human system. But that's just my guess. I don't actually know that. And then from there, the GRB2 SOS complex, and remember that the portion of the GRB2 where that arrow is, that whole purple thing is GRB2 or SEM5. Where the arrow's pointing would be better denoted as SH3, okay? There are different domains in there. If you guys look in the Vote, and Pratt book, it shows some crystal structures of that, but those are all, there's 2 of the SH3 domains and 1 SH2, and that whole thing is GRB2. And it's that the SH2 domain binds to the phosphate, and the SH3 domains bind to the SOS protein. That complex is a guanine nucleotide exchange factor, which is commonly denoted GEF, and that induces RAS to exchange its GDP for GTP. Cool? I found a decent video of this, because people talking about it typically doesn't, okay. Okay, I guess we're gonna have to watch this. Wait, let's go back for a minute then while we, oh yay, cool. Okay. So this guy takes a minute, but a good rule of thumb for the pathways, find a couple on YouTube and look through them. That link I attached should be in the one that I posted for you guys on, but it kind of helps make this a little more sensible. So when we see just the pictures, it's a little hard to see how things work. But what's going to happen is those two units of the RTK, whether they're already dimerized or not, are going to bind that, bind the ligand. And from there, the phosphate groups that are in the cytoplasm are going to be sequestered into phosphorylation of the tyrosine residues. This stuff. Person's talking on here, and I turned him off. All right, but they're going to autophosphorylate each other, one after another. Boom, boom. And then from there, the GRB2 and the SOS residues come on and they bind. Good, good. All right, and an important point to note is that as that SH2 domain binds, it causes a conformational shift in the SH3, which allows it to bind SOS. And from there, that guy will cause the exchange of that GDP with GTP on RAS. And then note that GRB and the SOS are cytoplasmic proteins. They're floating around in the cytoplasm. RAS is membrane bound, okay? So it is stuck up in the membrane. And then it's activated once it grabs that GTP. And I think that this is almost a point where the video becomes not relevant. Oh, we're still good. And then it's going to bind the RAF. And this guy causes a phosphorylation cascade. And what happens is that it uses ATP to phosphorylate each subsequent protein. It's not that it's passing along the phosphate group, it's that it phosphorylates each one using ATP, all right? And here's where we will stop because this is no longer important to us. But the part we need to know up to is that it grabs all these things, pulls them into the nucleus, and from there it causes some sort of gene expression, whether it be inhibiting a gene or activating a gene, okay? And that is that pathway. Are we all good with that? Yes? Cool. All right. The exchange of GDP for GTP causes it to bind RAF, and this initiates the signaling cascade. RAF is a serine-threonine kinase that once activated will phosphorylate a protein known as MEK or MAP kinase. Those are the same thing. Activated MEK will phosphorylate and hence activate a family of proteins known as mitogen activating protein kinases, MAPs, something, and the MAPs are also known as extracellular signal-regulated kinases, which are ERK. So MAPK and ERK are, from what I understand from the book, are essentially the same thing. I'm sure someone somewhere, there's a slight difference between them, but for the purposes of this class, we're going to consider them the same. And then MEK stands for MAP kinase, ERK kinase, activating kinase. Cool? And then the MAP kinases then move into the nucleus and phosphorylate a variety of proteins, including those three, and those are all transcription factors. Does that make sense to everybody? Thankfully, this pathway is pretty, it's unidirectional. There's no like branching or anything that we have to worry about. All right, and that's a quick summary of this, and this is posted online for you guys, so we're not going to go through this. Good? And there's another picture. If you guys follow that link that's up there, it takes, I think it's the University of Oregon, where I stole this picture from. This is the exact same thing as what your book has. It just has it denoted a little different in case that picture makes more sense to you. Good? Any questions on this? All right. We are done with this one. And we want phospho. I really hate this name. I can never pronounce it. It's phosphoinositide, I think. I just call it like phospho, and I just mumble the rest of it. All right, overview. Signal transduction generates a secondary messenger, and it is all triphosphate, which triggers calcium release, and generates formation of diacylglycerol, which activates protein kinase C. Calcium activates calmodulin, which activates and binds target. Calmodulin has, if I remember correctly, it like binds over 100 different proteins. So when calmodulin is activated, it goes after everything, and so it activates a ton of pathways within the cell. And then PKC and calmodulin activate proteins, and this will have an end result of a cellular response. Good. Ligand binding, same as normal. What's nice about these pathways is that because each of the proteins that are inside the cell can do multiple actions, we only need a small ligand to produce a large response inside the cell, okay? So for instance, I think in this one, it's epinephrine, and we only need one of those epinephrines to activate that protein, and that sends a cascade. And so if we have a bunch of these proteins on the surface of the cell, only a small amount of epinephrine is needed to produce a large response, all right? That first part is a heterotrimeric G protein. What happens is that signal binds, it activates the G protein, which causes that alpha subunit to come off, and that goes after the activated phospholipase C. So the formation of IP3 and DAG, diacylglycerol. The phospholipase C causes the PIP2 to be cleaved. I like this picture because it kind of shows you where those phosphate groups are. So the diacylglycerol is that thing up in the membrane. It has those two hydrophobic tails, and it's a phosphoglycerolipid. And so what's able to do is that phosphate group is bound to the sugar, all right? Phospholipase C goes and cleaves that, producing the diacylglycerol, which stays in the membrane and produces IP3. And the name of that stuff is inidazole 1,4,5-triphosphate. I believe for both lecture classes, you have to know the structure of this, yes? Dr. Kutsuli's, I know. Dr. Kopashkov's class, do you guys have to know this? Do we know if we have to know this? Check your guys' memorization list, okay? I know Dr. Kutsuli's class, you guys have to know the structure of this, okay? But it's just a 6-membered ring, and it has those 3 phosphates on there. The DAG portion, the diacylglycerol, remains associated with the membrane. IP3 is released into the cytoplasm, and it goes to the lumen of the endoplasmic reticulum. And from there, it's going to open a calcium channel. Is that calcium channel an active or a passive channel? Passive, right? It's just releasing calcium from high concentration to low concentration. At the end of this pathway, when it's signaled to put the calcium back in the endoplasmic reticulum, what sort of channel is needed to get that calcium back in? Yeah, the calcium ATPase, right? We need active transport to build that gradient back up. Good? So the IP3 binds to the calcium channel, it triggers it to open, and this releases calcium into the cytoplasm. Good? All right, so an increase of concentration of what compounds occurs shortly after the binding of epinephrine to its receptor in the phosphoinositide pathway. Think about that for a moment. Dr. Kutsuli's class, I can guarantee she was going to ask some variety of this in the form of, if this happens, what concentration is going to change, all right? And this tests whether you understand the pathway and you just haven't memorized a bunch of terms, all right? You have to understand what happens when certain things occur. A raise of hand, who could have answered this if it was on a test? A couple people, that's not enough, all right? We have our test coming up on Tuesday and Wednesday, yes? Shortly. All right. Answer, IP3 and calcium due to IP3 binding the calcium channel. So when that guy initially cleaves out, a lot of IP3 is built up. An important point to note, the DAG, diacylglycerol, stays associated with the membrane. So its cytosolic concentration does not change. All right? Its concentration in the cytosol remains exactly the same because it remains associated. Good? And then eventually, the inidazole polyphosphate 5-phosphatase will hydrolyze IP3 to increase the concentration of IP2, okay? So down this pathway, after a while, that enzyme will cleave, and so both IP3 and IP2 would increase. IP3 would increase first, and it would be degraded into IP2 over time. Good? Which would technically also increase the concentration of phosphate. Everybody good with that? All right. The fate of DAG, so diacylglycerol will diffuse through the membrane to associate with protein kinase C, also known as PKC. PKC can then activate other cytosolic proteins through phosphorylation using ATP as a source of phosphate. I should say phosphate ATP. In some cases, calcium also activates PKC, which wow, there are a lot of typos in this. Activates PKC, which comes from the ER. So remember that calcium that was released due to the IP3, that in some cases will also activate the PKC. Are we all good with that? A lot of typos. Oh, wait. Calmodulin activation. As the name implies, calmodulin requires calcium to work. It's a ubiquitous eukaryotic protein that participates in many cellular regulatory processes. The binding of calcium activates the protein. So what happens is that as that calcium binds, this protein opens up and it reveals this patch of methionines that is then able to bind to other proteins. And these other proteins all have an alpha helix that it has fairly basic residues, so that thing binds around it. Anybody ever had like a brace before? Or like seen, everybody knows what like an arm brace is? Yes. It's very similar to that in that it's going to essentially brace up that protein and it's going to promote it to work better, okay? So like if you roll your ankle or something, it's kind of hard to run. If you put a brace on it, it allows you to then run relatively well, provided that brace is there, right? Same idea with that calmodulin. It's going to enable that protein to do its job. When the calmodulin is not there, the protein cannot do its job. And so it's a control point of those proteins, all right? Once the proteins have calmodulin on them, they can then go and phosphorylate other things and again activate some cellular response, okay? We don't care about the cellular response for this class. We only care about how we get there. Oh, review of chapter 7. So this crystal structure of the rat testes calmodulin is shown. How many amino acids are in the alpha helix that connects the two calmodulin domains? And then I'm going to tell you the part that you need to consider starts here in green and goes all the way down to here in yellow, okay? But I want to know how many amino acids is that fragment? Take a moment and try that. Good. Let me ask you this. How many amino acids are in one turn of an alpha helix? 3.6. How many turns are in that particular alpha helix? Yeah, I think it's 7. I counted 7, and the book also claims it's 7. So we take 7 times 3.6. Some people say 3.5 or 3.4 or 3.7. Depends on who you ask, but 3.6, I believe, is the term it's usually used. But all those will get you about the same answer, all right? So we go through and do that. The alpha helix makes 7 turns. Each turn contains approximately 3.6 amino acids, and so the motif is about 25 amino acids long. Good. For Dr. Gutsuli's class, she usually asks a very similar question about contour length of either this or DNA. That means that you need to know how many how many amino acids per helical turn in a polypeptide helix, and you should also know how many base pairs in a DNA helix. Are we all good with that? And then just a note, it may be asked in terms of contour length, and that's just saying how long, essentially what is the distance from here to here, okay? It's not saying like how long is it around in the loops. That's not what it's asking. It's saying just measure this distance right here. Is everybody good with that? So you could back calculate or calculate the distance based off the 25 amino acids. Good? In many of the pathways discussed in this chapter, many of the steps require phosphorylation of proteins to allow the cascades to continue. What is usually the source of phosphate? So where does this phosphate come from? ATP. Common misconception. It does not transfer. Like it doesn't protein transfers a phosphate, okay? Those are fairly low energy compounds. In order to get this signal to amplify through the cell, you need a high energy source of phosphate, okay? And that's going to come from ATP in each step. All right, so this requires a decent input of energy. All right, ATP hydrolysis, the phosphates that are removed from ATP is placed on the protein. Good? All right, it's a hypothetical question, all right? I tried to make that very clear in here, but kind of a little bit of chapter 10, the membrane potential stuff. A high affinity calcium ionophore was artificially introduced into a cell. Assuming that the ionophore was soluble and functioning in the cytoplasm, what effect would it have? Any thoughts? What do ionophores do? They can transfer ions. Let's say we have a bunch of empty ionophores. What are they going to want to bind to? What are they going to want to bind to? Let's say I have an ionophore that is selected for potassium. What ion does that ionophore bind? Potassium, right? In this case, you were told that it's a high affinity calcium ionophore. Now, if we introduce a completely empty calcium ionophore into the cell, right, this stuff is going to start grabbing onto calcium. Now, once it's grabbed onto, that calcium is either going to dissociate or it's going to stay hooked there. And since we specify this as a high affinity one, it's actually kind of cool. There is a chemist here at UCF who works on this. But what they do is they use ionophores to capture ions. They use them for sensors. But once it's been captured, it can be quantified in solution. That's not important for this question, but just as a side note. So if we are losing calcium, if it's getting captured by that ionophore, what pathway is going to be affected? Yes, the one we just covered, right? The phosphoinositide, the one I can't say. That's going to be affected. That calcium, hypothetically, the ionophores would bind calcium, and if enough calcium was bound, that pathway wouldn't properly function because calcium is needed for the activation of both PKC and calmodulin, okay? Calmodulin needs that calcium to be able to work. So if their calcium has been sequestered by the ionophore and it's no longer there, these pathways are going to be hindered, all right? And that is that. Are we pretty good with these two? Yes, these are not the only two pathways. You should also know the heterotrymic pathways and the adenylate cyclase. I did not have time to go through and make a PowerPoint for that, so I don't have anything for you guys on that. There was a questions document posted that you guys in Dr. Katsuli's class would have known about. All right. That is that too, and we are currently all right, we have about an hour and a half left, so I'm going to open the floor up to questions. Do we have a particular topic that we'd like to cover? The multiple choice practice exam you may ask about. We're not going to go through every single question because that's already up, but if you guys have a particular question from it, I'd be more than happy to answer it. Yes, ma'am. Let's go biochemistry. All right, if you guys go down here to the amino acids page and you click on isoelectric point, it will pull up a page. This page does a fairly good job of explaining the concept of the pKa. This is something that people tend to not, it's weird, pKa. It is a quantitative measure of whether or not an acid will be protonated or deprotonated, okay? And it's weird to think about because it's not that it's changing at that point per se, it's that that's the halfway point. And usually what we say is plus or minus 1 pH unit on either side. That's where you have fully one form and fully the other. Are we all good with that? It's a good point to note that if you are lower than the pKa, it is protonated. Whatever it is, whether it's a carboxylic acid and it's amine, it'll be in its protonated form with whatever charge it has associated with it. If we go higher than the pKa, it is deprotonated. Is everybody good with that? This is always true, okay? If it's lower than the pKa, it'll be protonated. If it's higher than the pKa, if it's lower than the pKa, it'll be protonated, and if it's higher than the pKa, it'll be deprotonated, okay? And now the charge is compound dependent. So for instance, tyrosine, cysteine, and the carboxylic acids, those all will be neutral, but negative above the pKa. In the case of the amines like arginine, histidine, lysine, those guys, and the amines that are at the end of the polypeptide chain, those are positively charged because they're protonated below the pKa value, and they're neutrally charged above the pKa value. Are we all good with that? Yes? So let's say you have some protein, or some polypeptide I should say, and it has the sequence HCl. Cool? What two groups should we take into account that are on either side of this guy that people tend to forget? Yes, the N and the C terminals. Okay, those are important because they perform acid-base chemistry. We care about them. Okay? So we have this protein that has its terminals. I have a shorthand trick that I use. I know a lot of people will use the titration curve, and that's perfectly acceptable. It takes a little bit more time, and so I don't like that. But what I'm going to go through and do is, first off, I'm going to determine all the side chains. All right? At this point in the class, hopefully you have these memorized very, very well. Who's going on to Biochem 2? Okay, a decent number of you guys. Well, not decent, but a few of you. In Biochem 2, you do have to know the amino acids still, and you have to know them very, very well, okay? If you are, well, I guess that really is the only course that you would need them for at this point. But anybody who's going to the chemistry field, you should know the amino acids, all right? We have 3 residues on here, only 2 of which we're going to care about. Does histidine have a ionizable site on it? Can it gain or lose a proton? Yes, one of the nitrogens in that imidazole ring. And what are the possible charges on histidine? Plus or neutral. The same is true for the terminal amine. It can be either plus or neutral. What about the cysteine residue? Can that guy be ionizable, or is it ionizable? Yes, and what are its 2 possible charges? What? Neutral or minus. So when that proton gets pulled off, we have an S minus, that's sulfur. Yep. So this stuff can either be neutral or minus. Good. What about leucine? Do we care about that guy? Does it have an ionizable site? Nope, not one that we care about. So I'm gonna ignore that guy. Carboxylic acid terminal, what are its 2 possible charges? Good, neutral or negative. On exam day, you guys will have a table that you can go look these things up in, but what you're then going to do is you're going to find each corresponding functional group, and you're going to assign it a pKa. The N-terminal is usually about 9. Histidine is 6.04. Cysteine is like 8.4. And then the carboxylic acid N 3. Good. And again, on test day, you would go look those up in the table. There's no need to memorize those. All right? From here, what we're going to do is we're going to count up the max number of positive and the max number of negative charges. And it looks like there are 2 positive charges and 2 negative charges. And so I'm going to make myself a number line of sorts here. That essentially goes from the max positive to the max negative. Are we all good with that? From here, this is important, okay? If you are messy, don't do that anymore. This has to be very clean. The line that I'm going to draw goes between the charges, not on the charges, between them, okay? This is very important. They go between the charges. Good? The number of lines should correspond to the number of pKa values that you have. We have 4 lines, we have 4 pKa values, so it looks like we're good thus far. What I'm going to do is I'm going to order them from smallest to largest, all right? And that should make sense. At +2, we're fully protonated, meaning that which pKa should we encounter first if we're moving up in pH? The carboxylic acid one, right? We should hit the lowest one first. So that's going to be the first one we come across. We would go through and put these on here. And now what we have is the ability to determine at what pH the protein, or in this case, the rather small polypeptide, is going to be at any, what charge it will be at any pH. Are we all good with that? Yes. It will be at pH of 7-ish. It will be deprotonated, and histidine will also be deprotonated. But the cysteine and the nitrogen will still both be protonated. Is everybody good with that? Yes, ma'am. It has the lowest pKa value. You just take the pKa's and you put them in order from lowest to highest. It just so happened that this one was the lowest. And in most cases, either it or glutamic acid or aspartic acid will be the lowest. It just depends on what sequence you're given. Good? And now from here, it does not matter. If we're asked for the PI, that's just a euphemism for what is the pH when it's 0. All right? We would take the 2 values on either side of 0 and average them. And in this case, since it's like 6.04 and 8.04, that's probably going to be about 7.2-ish, somewhere in there. Somebody do that on a calculator and tell me what that is. Cool. All right, so it's about just over 7. If let's say I wanted pH at which the protein was net +1, well, I would go to +1, and I would take these 2 pH values, and I would average them. And it looks like that should be about 4.5. Does everybody see that? So we would take 3 and 6, and we would average it, and that 6, 7, 8, 9, yeah, 4.5, right? Does that make sense to everyone? So now not only can we find the PI, but we can also find the pH at which it is any charge. Does that make sense to everybody? Yes, hopefully. All right, that same process will be true of every protein. I shouldn't say I say polypeptide. These are not proteins. But we're going to go through every ionizable group. We're going to decide if it can be plus or neutral or minus or neutral. We'll assign the correct pKa value. We're going to go through and make our table where we start at the most positive and go to the most negative. We'll put a line in between. Note, this is not on. This is in between each charge. If you put them on each charge, you have a very high likelihood that you're going to mess up when you pick the two charges. All right? Because when we go for 0, we want the pKs on either side of 0. Not on 0 and one below or one above. Is everybody good with that? Yes? Any questions on this? Yes, ma'am. That is by knowing the functional group. So you have to have these things perfectly memorized to be able to do that. So for instance, histidine has that amine group on it, and when that's protonated, it'll be positive, and when it's deprotonated, it'll be neutral. Yes, yeah, you look at the side chain. Like cysteine has the SH group on it. That can lose a proton or have its proton. When it has its proton, it's neutral. When it loses its proton, it's minus. And a good rule of thumb is that the carboxylic acids, tyrosine, and cysteine, these will all be neutral or negative. Anything with an amine or a nitrogen in it is going to be plus or neutral. That grabs everything. So if it's got nitrogen in its side chain, so arginine, lysine, histidine, the N terminal, those are all N, they're nitrogen, so they'll be plus or neutral. Anything that is not that, essentially, so the carboxylic acids, tyrosine, and cysteine, those will all be neutral or negative. Did I miss any there? I think that was all the ionizable ones, yes? Good? Does that make sense to everybody? All right, next request. What would we like to do next? Yes, ma'am? Yes. Okay. By raise of hand, who was in lecture for Dr. Katsuli the other day when Riley came in? Yes? Did we all remember the columns that he brought in? Yeah? Column chromatography is a very common practice within biochemistry. Biochemists always need to be able to separate stuff out. And the way that they do that is through column chromatography. And some really smart people a really long time ago figured out the specifics of all of this, and they were able to say, okay, if we use this substance within the column and we pass X, Y, and Z through, we can get these guys to separate based on either polarity or charge or some other factor. There are some really cool ones that actually do chirality. So you can have like columns that separate chiral molecules, sort of like hold on to the R ones and release the S ones, or DNL, whatever one you're interested in. And we have 3 types that we care about in this class. We care about cation exchange, anion exchange, and reverse phase hydrophobic. And the way I remember these is that whatever the name of that thing is, whatever it's called, that is the thing that it holds on to the longest, and it spits everything else out that is not what its name is, okay? So a cation exchange column holds on to what sort of compounds? Positive, cations, right? Anion exchange holds on to anions, and it releases everything that's not an anion. And reverse phase hydrophobic holds on to the hydrophobic. I believe that's also referred to as just a hydrophobic column. I don't know why that reverse phase is in there. It's beyond what I know. But the hydrophobic column is going to hold hydrophobic things and release more polar molecules. Are we all good with that? Yes? These guys are pH dependent. So for instance, histidine, if we have histidine and lysine, these guys both have an ionizable site that's positive, yes? And depending on what pH it is, we can either make them the same or different. If the pH is below 6, if it's below 6, these are both positive. If it's above 9, but between 6 and 9, we have histidine will be neutral and lysine will be positive. Does that make sense to everybody? All right, and that's based off the pKs of their side chains and assuming that the N and C terminals are approximately the same, have approximately the same pKa values. Good? So if I were to run a column on these guys, whether it be cation or anion exchange, it would not matter. But what pH should I use to run this? Should I use 5, a pH of 5? Should I use a pH of 10? No, because they're going to have the same charge, right? And while there may be slight separation, there will not be a large amount of separation. And for column chromatography, we want large separation so that we can collect the what's called the elutant and then remove that container that we caught it and put another container underneath and collect the next one. I'll tell you from experience, running a column is very boring. You just have to sit there and watch it. Most of the time what you have is not colored, and so it's really boring. Is anybody taking who's taken the first organic chemistry lab? A couple of you guys. Is anybody taking the one that's after that? It's like 3215. A few of you guys. You guys in your first lab will run a column, and it's spinach, but what you do is you separate, and you get to watch all these really pretty colors separate on the column. It's actually quite cool. But for this guy, we'd want to run it somewhere between 6 and 9. I believe both on your first exam in Dr. Katsuli's and on one of your quizzes for Dr. Kopashnikov's, you guys had a question about which column to use to separate histidine and arginine. Yes, do we all remember this question? Hopefully. It was almost exactly like this. And what you would do is you would pick either column, all right, anion or cation exchange. It usually does not matter as long as the pH is correct, okay? So for this one, I would probably pick cation exchange, and I would run it at a pH of about 7.5 to 8. Why did I pick 7.5 to 8? What is special about that pH? What? Beautiful, right? And we want to find the maximum distance from both. If we get too close to 9, a lot of this stuff will be deprotonated, all right? And that can be, we can figure that out, calculate the fraction of that that's going to be deprotonated. And if we're too close to this, some of this will still be protonated. Because remember, pK is not an absolute value. It is it's the halfway point. And so we have to go a full pH unit past the pKa to be almost entirely deprotonated or protonated, all right? And we can do calculations with that. Let's do that real quick. I guess let's say with this board. So let's say that I have histidine, and we know that its pKa value of interest is 6.04. And I want to know what fraction is protonated at a pH equal to 6.4. Good. Take a moment to try this real quick. For Dr. Kopashnikov's class, he loves this question. I believe it is on your sample final. If I remember correctly, it was on our actual final. So you need to be able to calculate the fraction that is protonated. For Dr. Katsuli's class, she usually asks this in a manner saying, what relative ratio or what percent will this stuff be given this pKa and this compound? Or she might ask for the pH given the concentration of log and base, okay? Let's take about a minute. Everybody try this real quick. If you need help, call me over. But I'm going to give you guys about a minute to try this and work it out. Yes. Well, it depends on the particular compound. So if you have a lot of stuff that's negative, you might want to go with an anion exchange because you know that it'll hold on to the anion ones really well. In this case, because one is positive and one is neutral, I'd go with the cation because you want it to hold on to the lysine for a while and release the histidine. But you ideally want to pick the column that's going to hold on to something tightly and release everything else and then release that compound. For the pKa value, if the pH is below the pKa, it's protonated, and if it's above the pKa, it's deprotonated. So pKa is just the measure of where that deprotonation occurs. Good. Has everybody attempted this? We can do this. Yes, no, maybe so. Let's try this. So we know that its pKa value, we're given a pH. We're just going to plug this into the Henderson-Hasselbalch equation. So we're going to say that pH is 6.4 equals pKa plus log of base over acid. We'd move this term over, and we would get 0.36 equals the log of base over acid, and base over acid is going to be equal to 10 to the 0.36. Can someone do that real quick on their calculator? What is that value? 2.29. Beautiful. So it's going to be 2.29. Us is that for every 1 molecule of acid that there is, there are 2.29 that are in the basic form. They're the deprotonated, all right? And we can put this into a relative percent. So what we could say is, like, say we wanted the percent that was acid. So what we would do is we'd say 1 divided by 3.29 times 100%. So if we wanted the percent that were in the protonated form, we would divide that. We'd get like 30%-ish. Yes, 29 maybe. 3.29. We're doing a fraction. Remember, if someone asks you what you got on a test, do you say how many I got right divided by how many I got right? Does anybody do that? I hope not. You say how many you got right divided by the total, yes? Well, what we're saying here is how many are protonated divided by the total, and the total is 1 plus 2.29, because it's approximately 3.29 molecules. Does that make sense to everybody? Does that not make sense? Anyone need that explained further? All right. Are we good on this? Did that answer the question about the column? This can be applied to more complicated polypeptides. The theory behind it is still going to be the same, right? You determine where they're going to have a different charge based on pKa values and your knowledge of pH. And then whatever column you pick is going to be, or whatever column you're given, it's going to hold on to its, I always think it's going to hold on to its name the longest. So if it's a cation, it holds on to cations for a long time and releases everything else in order of whether or not they're an anion or a cation. Does that make sense? Yes? No? Maybe so? Good? All right. Next topic. Yes? On the multiple choice or the F response? The multiple choice. Presentation. I totally spelled that wrong. Okay, so this says calculate the free energy required to move 1 mole of sodium ions from outside the cell where the concentration is blah, 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 to inside the cell where it's some other number. When the membrane potential is 60 millivolts and the temperature is 37 degrees. Okay, let's hope this isn't one with an error. Was this you think there's an error, or was this just how do we do this? Okay. Let's hope this turns out a typo here. So for membrane energetics, there's an equation we need to know, and that's going to be that delta G equals RT natural log of in over out plus ZF psi, and that should be delta psi. Cool? Now, I always forget all the sign crap that goes with this, and so instead what you should do is you should kind of have some logic to it, all right? There are 2 terms to this. There is a concentration term. This tells us the free energy relative to the concentrations, and there is an electrical term, and this tells us the free energy required to move that electrical component, okay? And some things that we need to know is we need to know that a cell is net negative inside and net positive outside. Are we all good with that? And if we know that, we can predict which direction the electrical term will be favorable. So if we have a positive ion, which way is favorable to move? In the cell or out of the cell? Electrically. In the cell. And if we have a negative ion, which way is favorable? Out of the cell, right? And so when we do these calculations, what we can do is we can make a prediction about which term is favorable based on the concentrations and which term is favorable based on the electrical part. Is you don't have to necessarily remember the signs. And I know that I never do. I always forget. And so what I say is, okay, well, what is favorable in terms of the concentration and what's favorable in terms of the electrics, and we can make a determination of the sign, because we know that negative values are a spontaneous or a favorable delta G, and positive values are a non-spontaneous delta G. Yes? All right. So it says, what's the free energy? And it gives us some concentrations, and it gives us a temperature. So I'm going to say that this is 8.314. The temperature is going to be 310. I'm going to take the natural log. And now in this case, I'm going to say 160 divided by 10. Because these are both in millimolar, and because it is a 1-to-1 ratio, we don't have to change the units. If that is not true, if that were like ADP times PI divided by ACP, you have to put it into molar. I repeat, if it is not if it's not a 1-to-1 ratio here, if there's any exponents, if there's another term, you have to put it into molar, okay? Those units won't cancel, you'll get the wrong number. Are we all good with that? Yes? When in doubt, convert to molar, okay? Don't not convert. This term is going to be our gradient term, and so what we're going to do is calculate this. Can someone tell me what this value is? Yes? We're going to get there in a minute. For now, because the magnitude is going to be the same. So what's the magnitude of this number? Someone with a calculator. If you can't do this, test day will not be fun, I promise. Good. And then can someone tell me what the electrical term is equal to? Who has the concentration term? Can you do me a favor? Can you divide that by 1,000 and put it in kilojoules? Whoops. All right, and what is the electrical term here? So it's essentially 96,485 times 60 divided by 1,000. And then divide it again by 1,000 to put it in kilojoules. Yes. What? That's why I divided by 1,000. Yeah, so 60 over 1,000 puts it into volts. When it's in joules or kilojoules? Divide it again, because we have two sets of, so it's going to be like about 5. Oh, wait. And now what we're going to do, for the time being, I'm going to put these plus minus. Let's say on test day you forget which sign goes where, and I do that all the time, so we're going to talk about this. Let's start with the concentration term. The question says to move sodium, which is a positively charged ion, well, we don't care about that, from outside the cell, where it's 160, to inside the cell, where it's 10. Going from high to low concentration, is that favorable or disfavorable? That is favorable. So should this have a negative or a positive sign? This should have a negative sign, because concentration-wise, this is a favorable process. Cool? Now let's talk electrically. Sodium is a positively charged ion, yes? So for the electrical term, if it's moving, and remember, we have to know this about the cell, that it's negative inside, is this a favorable or a disfavorable process? To move a positive inside the cell. Favorable, right? So this term should also be negative. Is everybody good with that? Yes? And then it looks like we should be getting about negative 13-ish. Is that like negative? Did I do that right? Yes? And that looks like answer choice C. Did that make sense to everybody? Good? Any questions on this? Yes, ma'am. Well, to me, I can never remember which one to use. And since the magnitude of this number will be the same, whether it's in or over in, what we decide at the end is once we have this magnitude, we can look at the question and see where the high and low concentrations are, and we can determine the sign that way. Since the magnitude of that number is going to be identical. If you had done the opposite, if you'd done 10 over 160, you would have gotten this number off the bat. We did 160 over 10 and we got positive, but what we were then able to do is look and say, well, it's moving from high to low, and that is spontaneous concentration-wise, and so that term is negative. And then we looked at the electrical and we did the same thing. We said, okay, we have a magnitude here, and since this is moving from a positive to a negative, and it's a positive ion, this would be favorable. Had this been, say, chloride, let's say it were chloride and the concentration was the same, this term would still be favorable if the concentrations were the same, but because chloride is negative, this term would have been unfavorable, and we would have made this positive. Does that make sense to everybody? Yes? All right, what next? In, over, out, yes, I believe so. Yeah, 10 over 160, that'd be negative. Yeah, if you plugged everything in as that equation is written, you should get the right thing. Yeah, so I flipped the concentration terms, but that was mostly just to make a point that you don't need to have it memorized. All right, if you guys go for the logic of it, of why, like what each term is, then you can make a good, you can make an assumption about whether it's going to be spontaneous or not. Good? All right, what next? Yes. Is this in the multiple choice? I don't know. Let's look. Wait, which, in this packet? And which number am I going to? Chapter 14. Consider the following metabolic reactions. Acetyl-CoA. Yes, I think there might have been a typo here. Let me double check. It should be negative 32.65. Yeah. Do we want to work that one out? Okay, let's do this. It makes you guys feel any better. The first time I got this question, I got it wrong. All right, so the question is saying, consider the following metabolic reaction. We have succinyl-CoA and acetoacetate make acetoacetyl-CoA and succinate. And it's giving us the delta G for the hydrolysis. So we have a couple reactions shorthand some of this to make it a little easier to see. But we have succinyl-CoA plus acetoacetate reacts to form A-CoA plus succinate. Cool. It should be AA-CoA. Cool. And it's telling us that this delta G value is equal to negative 1.25. And then it's saying for the hydrolysis of succinyl-CoA, so it's saying that we have S-CoA plus H2O reacts to form succinate plus coenzyme A, and the delta G for that guy is negative 33.9. Good? And then it's saying, what is the delta G for the hydrolysis of acetyl-CoA? So it's saying AA-CoA plus H2O reacts to form AA plus coenzyme A. Good? And it looks like as written, G, we'll call this 1, 2, and 3. And that's the delta G that we're looking for. And it looks like that if we add these bottom two equations together, we can get that equation. Does that make sense? So we're going to do a little bit of rearranging. And I think that I'm going to flip that guy. And so we're going to have coenzyme A plus acetoacetate reacts to form coenzyme CoA plus H2O. And that's going to have some delta G value associated with it. And then we're going to say that this guy is the same as written. The coenzyme A will end up canceling when we go to do this. The waters will cancel, and we'll get that final equation. Because this one remained the same, its delta G is the same as written. Negative 33.9. Good? And then we're going to get a final equation that's going to say succinyl-CoA plus that guy reacts to form. And that has a delta G value of all right. And let's bookkeep and make sure everything was done right. Succinyl coenzyme, acetoacetate, acetyl, good, good. Oh, or do that. That's no. All right, whatever. Oh, and then we're saying the delta G for the hydrolysis of succinyl-CoA. So when this guy is reacted with water to form this, and that should make sense. Hydrolysis reactions of biological compounds typically produce free energy because it's an increase in entropy. And they want to know what is the delta G of hydrolysis for this guy. So what we're going to do is we're going to determine the delta G for this one. And delta G3, it's going to be delta G3 plus delta G2 is going to equal delta G1. So I'm going to say X minus 33.9 equals negative 1.25. And I'm getting X equal to positive 32.65. But what do we need? This is for this guy. Is that, that's not written right. Haha. It should be, oh yeah, it is written. For this guy, is that the hydrolysis? No, the reverse is the hydrolysis. So the delta G value that we got down here, that positive 32, we need to flip the sign because when we flip that equation, that's the hydrolysis reaction. So our end result should be negative 32. And that should make sense. Apparently I wasn't thinking that way when I took this quiz forever ago, but a hydrolysis reaction of a biological compound is going to almost always release free energy. So the hydrolysis of ATP, the hydrolysis of any of these coenzyme compounds, the hydrolysis of any of the nucleoside triphosphates or nucleoside diphosphates, all those hydrolysis reactions will produce energy, and so their reaction as written forward will have a negative delta G. Can you see all that? Do you have any questions on this? Yes, no, maybe so? Good? All right, what next? Saw a hand over here earlier. Disappeared. I have no thoughts. No questions for me, Victoria? You're looking, ma'am. So it's in the review portion of the multiple choice. Where did, oh, there. This guy? This one. All right. So it says, what is the value of Kd given the fractional saturation of a sample is 5% and the pressure of oxygen is 36 torr, and that the value of the Hill constant is 2. Let's sidetrack for a moment and talk about this. What time is it? 4:45, and we are finished at 5:30, yes? Okay, so we still have a decent bit of time. The Hill equation is one that everyone's going to need to know. You will have some variation of it likely on all of your finals. And there's a couple things that we need to know. All right, KD is the same as saying P50. KD is equal to P50. All right, and that's just saying at what pressure is this guy 50% saturated, okay? And KD is something you can get for a lot of proteins, right? It's not just hemoglobin or myoglobin. KD can also be found for a receptor within a membrane, right? If that's 50% bound, it's reached its KD, yes? These guys all have very similar looking graphs. For myoglobin, it's going to be that hyperbolic looking thing, and for hemoglobin, it's going to be that S looking thing. So we have myo on top, hemoglobin on the bottom. Remember that hemoglobin is a sigmoidal or an S shape. Myoglobin is a hyperbolic shape, and it kind of looks like a sideways, there's a word I want to use, parabola, that thing, sideways parabola. All right, for Dr. Gutsuli's class, I know that she'll often just ask you to describe the graph, like what shape it is. All right, we should be able to do that, yes? For both of these, we determine P50 by going to the point at which it's 50%. We track our way over, and then we track our way down depending on which one we care about. And whatever pressure that is, that's the KD. In this case, which one has a higher affinity? Myoglobin, right? Because at a lower pressure, it reaches its fractional saturation of 50%, okay? So it has a higher affinity for oxygen. There's an equation we can use for this, and we can say that the fractional saturation is equal to the pressure to the N divided by pressure to the N plus P50 to the N. All right, and it's going to be the Hill constant. Way back in the day, Hill thought it had to do with how many things it bound. That's not quite true, but it does give an answer. For hemoglobin, you're usually given a Hill constant or asked to find it. Note that if it's not given to you and for some reason you're told to guess, usually people say 3. Okay, hemoglobin usually has a Hill constant about 3 for normal old healthy people. That Hill constant changes if you have a certain sort of blood disorder. There's like, I think, close to 1,000 different mutations that can occur in hemoglobin, and it results in a whole variety of Hill constants. For myoglobin, that Hill constant value is always 1, and that's because when myoglobin binds to oxygen, it does not have any cooperativity to it because there's only one binding site, okay? Hemoglobin has a weird value of N, all right, aka greater than 1, because when it binds to oxygen, that binding induces conformational changes in the other binding sites, and these result overall in a different desire of that binding site to bind oxygen, and that's going to be expressed mathematically as N. Are we all good with that? Yes? Any questions on this so far? All right. Don't forget, as soon as we hit 5:30, the last time you will ever see me up until test day for Dr. Gutsuli's class, okay? So if you have questions, you must ask them now. Otherwise, I will never see you again. Yes, sir? Yes. Is that a hemoglobin, myoglobin? Yes. Yes, we'll find this, and that'll be next on the list. Cool? All right, any questions on this? All right. All right, for that question there, it's giving you a bunch of these numbers. You need to know this equation, all right, be able to plug and chug. The only other thing that you really absolutely have to know for hemoglobin, myoglobin is going to be the linear Hill plot. And really, this is just going to be reading a graph. All right, for the linear version of the Hill plot, there's two things you need to know to be able to get all the information out of it. The slope is equal to the Hill constant n, so you do normal delta y over delta x. I usually like to use intercepts when at all possible, just because that makes math easier. And then the x-intercept, not the y-intercept, the x-intercept is the log of P50. So if you find the x-intercept and you undo the log, so what that really means is that you say P50 is equal to 10 to the x-intercept. Okay? 10 to the next x-intercept is P50, and you're able to find P50 based off those two. And once you have P50 and N, you can essentially do any calculation you want. Cool? So be able to use this formula, know the shapes of these graphs, know that KD and P50 are interchangeable for the purposes of our discussion, and know the hill graph here. I think for Dr. Boshnikov's class, you guys got like shrimp, like a shrimp one, right? And it was like weird values, so you couldn't memorize anything. Does everybody remember what I'm talking about? Yes? I don't work with that class, so you should know better than me. Everybody good on this? All right, let's move on. We had an oxidation reduction. Where is this question at? In her final exam? Oh, okay. Let's go dashboard. Yeah, for Dr. Kasuli's class, this is the final exam practice that she posted for you guys. Download this. And what number is it? 10. Oh, great, this one again. All right, let's do this. All right, so it says given the standard reduction potential of oxaloacetate is point whatever that is, and it says that the standard reduction of NADA is that we want to calculate the delta E for that reaction given. For these guys, I always make the mistake of trying to do them in my head. Don't do that, you'll mess up. Always write these out. So it's saying given the standard reduction potential of oxaloacetate, and when we write these, we should know that the way they're written is the oxidized compound plus electrons makes the reduced compound. And there's usually some stoichiometry there that for this we're going to ignore. But what we can say is that malate, no, I want to go the other way. I want to go oxaloacetate, and I'm going to call this OO, plus some electrons, reacts to form malate. Good. And it says that as written, that has a standard reduction potential, an E value, equal to negative 0.166. Good? And then it says that NAD reacts to form NADH. And its standard reduction potential is negative 0.315. Good? And now what we're going to do is we're going to flip these equations to get them into the correct format. So I'm thinking that this equation needs to be flipped. And so it's going to be malate reacts to form oxaloacetate plus electrons. And that would mean that its delta E value changes in sign, that's positive 0.166. Good. We're going to add these guys together to get our final equation. And so delta E is going to be positive 0.166 minus 0.315, and that should equal negative 0.149 volts. Somebody do that math to make sure that that's actually true. Is that good? Yes? The nice thing is that the delta E values, we're going to do the exact same way we did those delta G values, all right? We give them as written. Remember that oxidized plus electrons forms reduced, and that's that standard, that's the value that's given. When it says the standard reduction potential is blah, 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 that value corresponds to the oxidized plus electrons form reduced, okay? That's how that whole table is written. So even if it doesn't explicitly tell you that it's oxaloacetate to malate, we know that because what they're saying is we can determine that from the final equation. If NAD plus is the oxidized form, what does malate have for that to react? Yeah, it has to have the electrons, right? All right, if it doesn't have the electrons, it can't, those two won't react together. Does that make sense to everybody? Yes? And again, the same as we do delta G. Yes, ma'am? Oh, wait. Does anybody have any other questions on this one? Yes. Yep, yeah, they told us with the final equation, and then we do that, we'd get malate and NAD plus on this side, and this should technically be 2. There are 2 electrons for that, but that doesn't matter here. And then we know we want oxaloacetate and NADH on the product side. Yes? I flipped the equation. Remember, for delta G and for E cell, we changed the sign when we flipped the equation. Like when you flip in for this, because we were given these 2 as written, but we want that final one. And so that means we have to flip this one. And so I flipped it to form this guy. And I changed the sign of the E cell. Does that make sense? Okay. Any other questions on this one? All right, what question would you like? With a what? Nernst equation, yes. Okay, if you guys are in Dr. Kapashnikov's class, you can cover your ears and drone this out for a little while, because you guys didn't cover metabolism, right? Yes? Yeah, you guys got lucky, right? Because he forgot there was no class on Monday. Yeah, poor Dr. K. He was probably so excited, and then the school ruined it for him. Okay. The Nernst equation. The Nernst equation has a shocking resemblance to the free energy equation. Very similar. We have our E standard state. That's usually the one that's given to us. We have the E non-standard state. That's usually the one that we're looking for, or we're given that. We have R, which is going to be the gas constant, 8.314. It's the only one we use in this class. Temperature. Should I ever plug Fahrenheit or Celsius in there? No. At this point in our chemical careers, we all know to use Kelvin, yes? Always use Kelvin, all right? Kelvin's going to be the thing we plug in there. N is going to be the moles of electrons. A good rule of thumb for this class is that it's almost always 2, okay? Most biological reactions move 2 electrons at a time, all right? Now, the real answer is you should look at the chemical equations and see how many electrons are being moved around. But if you forget that, plug in 2, okay? Now, again, I'm going to tell you that's not always true, but it's usually true. You good with that? And then F is going to be Faraday's constant, which you should be given. And that's about it. And then this is going to be same old, products over reactants for the equilibrium constant. Good? I think that's usually given as K. Let me see if I have an example of this. This one I'm not so good with making up ones off the top of my head. Huh? 15. Technically, no. That is an example of delta G equals negative NF delta E. Like that's interconverting between those 2. Did you have a particular example you wanted to work? Does that answer the most of your questions? Yes. So let's I don't have any examples, but let's talk through this. This guy you would find like you would with normal. The E cell you would find by looking at the 2 equations. You got a value for that. R, T, N, and F are always going to be some sort of constant relative to that equation. So T is a temperature you're given. F and R are constants that you're given, and N is going to be electrons, yes? This is the exact same L and K, all right? Equilibrium constant, plug it in, do what you always do. And then that is it. This is just like the only major difference between these 2 equations is that moles of electrons times Faraday's constant. Does that make sense? Yep, yes, it would be exactly the same setup, just different. You'd have those 2 extra terms there, but just plug and chug, all right? There shouldn't be anything overly complicated. For Dr. Gutsuli's, make sure you guys know both these equations. For Dr. Kapachnikov's, make sure you know the second equation, okay? All right, next request. Yes, ma'am. Yes. Yes, you said number 5? Where did that go? Chapter 14, number 5. Okay. Consider the following metabolic reactions. Succinyl-CoA and acetoacetate react to form acetoacetate coenzyme A and succinate. Its delta G value is given. It says, what is KEQ for this reaction at 25 degrees? Please let this not be a typo. That's always not fun. There are a couple other equations that you guys should know to convert between F delta E. And we should know that delta G prime. Good. These two, if you know both them, you can interconvert between K and E and G and E and G and K or whatever it is you so desire. Remember that these are all in standard state, and that is K is in the equilibrium constant. Right? It's not Q is in wherever the heck it happens to be at, okay? That is K is in equilibrium. For this one, we're given delta G, we're given temperature, we're looking for K, so I'm thinking it's this guy. So I'm going to say negative 1,250 is equal to negative 8.314 times 298 times the natural log of K. I'm thinking that K is going to be equal to E to the 1250 8.314 times 298. Yeah. So I'm going to say K is equal to E to the, and I'm going to have divided all that stuff already. Does that make sense? Does anybody know what you get when you plug all that in? E, 1.66. Yes, everybody again on their calculator? Does that answer your question? Beautiful. All right, next. Yes? Mm-hmm. All right. All right, so to make my life easy, I'm going to pick the smaller one. There are a couple ways we can access. So for chain terminator gel, if you're in Dr. Katsuli's class, I can promise she's going to ask this in some variety. She loves this question. And why do I know this? Because she told you multiple times in lecture, right? If you don't know this, shame on you. Show up to lecture. It's early, but the rest of us have to. All right? She told you guys multiple times that this was going to be on your third test and that this would also be on your final, okay? She has promised it, so we can expect it. When we run a chain terminator gel, we kind of want to know the process a little bit because that can help us get the answer. We have some DNA fragment that runs 5' to 3'. Cool? We take that fragment and we want to analyze the sequence of the nucleotides. We want to know what order and how many of each nucleotide are there. And the way this is done is they come up with some primer, and usually what they do is they analyze just a small portion of this end, and they get the identity of the end fragment. And what we can do is we can label it with a primer. Cool? And remember, this stuff anneals anti-parallel. All right? So this stuff anneals anti-parallel. So we have a primer that we've developed. And then what we do is we put this into a bunch of different reaction vessels. And note, there are lots of these molecules. Not just one, there's lots of them. And we put a little bit in each reaction vessel, and we run it with DNA polymerase and some nucleotides. One of the nucleotides that we introduce is a dideoxy, meaning that it has neither a 3' nor a 2' hydroxyl group, okay? Which means that it cannot be joined to the next fragment, and so it terminates, hence chain terminator, all right? And what happens is that each of these nucleotides get added until we get to this 3' end. Well, in each reaction vessel, in one we do the G, in one we do C, in the other we do T, and then we do an A. And then we run these guys through an electrophoresis, they're going to show up as bands. Is everybody with me so far? Yes? When we run this guy, what we get, right, and in whatever order they happen to run those columns in, we're going to get this gel, all right? And we're going to use some sort of technique to allow those bands to show up. Is everybody good with that? And so maybe we get like this guy. Cool? And when that was done on a gel electrophoresis, what we know is that all the really heavy fragments get stuck up here. Meaning that there are a decent number of these that are all the way done. Yes? And there are some that are almost done, and there's some that are not really done. There's some that aren't done at all, right? The ones that aren't done at all are the lightest, and so they will travel to the bottom fastest. And what we know is that within every sample of this, there is going to be DNA with the primer only attached to it. And those will be the 4 bands down here. Are we good with that? Now, the ones that this is showing, this C means that we used a dideoxy-CTP. So that means that this band is a C, which means that this, whatever this fragment happens to be, that is a C. Does that make sense? And then we know that because this is the heaviest, what end of the chain is this part on? 3 or 5 prime? 3 prime, right? And we read down this, we're going to go C, C, T, G, A, T, and then this end is the lightest because it has just the primer, and so it's the 5 prime. Are we good with that? Now, depending on what the question asks, if the question says, what is the sequence of this particular fragment, all right, we would read this way, and everything up to that point, that's that fragment. But most times the question comes in the flavor of saying, what was the sequence of the original strand? Because that's what we care about. When people run this experiment, what they're trying to find is, what was this sequence? Is this the answer? No, we need to take the complement of that. And then remember that this 3' end is this 3' end, which means the 5' end is up here. And we read down this, it's going to read G, G, A, C, T, A. And this would be our answer. Does that make sense to everybody? Yes? So when we read this, we're going to take this guy, take the complement, 5 to 3. From the top is 5, bottom is 3 on our parent strand, or the initial strand, and that's usually our answer. The other way this could be asked is that this piece could be given, and then you're given the gel, and it asks for the primer. That's going to mean that what we did was we're given this, we're going to find how this part anneals on, and then whatever part is empty, that part is our primer. Does that make sense to everybody? If you guys look at this document on my website, there is a whole page of explanation about that, okay? It goes into detail, shows explicitly, like highlights everything, okay? So when you guys get a chance, go through and read that, all right? But for most of these, we just read across, and we're good to go. Cool? As a side note, for Dr. Katsuli's class, make sure you guys can do the PCR. Dr. Kapashkov also usually asks about this. I'm not sure if he would on your final, and I don't remember if he did when I took the class. But for Dr. Katsuli's, I can again promise, because she said it in lecture, and we should know this because we've been going to lecture at 7:30 a.m., right? Yes? The PCR is one that we have to know about. I will get back to you in just a sec. Yes? Yes, we can. What was your question real quick? Okay. What time are we at? 5:07. Okay, we have a little bit of time. All right, let's do a PCR real quick. Oh. Yeah. Oh, wait, I think I have one already. Let's do that. So it says, number 5, given the sequence of give the sequence primer for the following DNA strand, it would be more correct to say that it should give 2 for the strand given. Because when we're given a strand of DNA, it's usually safe to assume that it's in that the complement is also there. DNA prefers to be in the double helix. For the primer, which way does DNA polymerase go? How does it synthesize? Yeah, it reads 5 to 3, right? Which means that the primers that we need are going to be the 5' to the 3' of that little primer, and that's going to anneal on. All right? So if we have any given fragment of DNA written as such, what we know is that one of the primers that we want is going to be that, as written. We just take those first 10 or 12 or 15, however many you're asked for. We take those as written. That's our primer. Everybody good with that? The next one's a little more complicated. This primer anneals to the fragment that's not shown. Are we all good with that? This fragment is written. The primer that anneals is going to be the complement of this piece, and it's going to be backwards. So we would take that fragment, and now on a test, that would be written like you would physically flip that around and it'd be written the other way. Does that make sense? So for instance, the one on the board, or the one up on the screen, my primers would be G, T, T, G, C, C, G, A, 1, 2, 3, 4, 5, 6, 7, 8, 9, A, G. So it's a 10. So that's one primer. Cool? The other primer, we're going to read from the opposite end, and we're going to take the complement. So the second primer that we would create would be C, G, A, C, T, A, G, C, 1, 2, 3, 4, 5, 6, G, C, A, T. Cool. There. All right, and that one's a little trickier because we have to go from the back when we take the complement. And remember, you physically flip that thing over. Yes? Let's look at that. We're on the final exam. And then what question? 35? Okay, so it's giving us this. It says, what is the product of the PCR reaction of the double-stranded DNA below and the two primers? So we're going to first look for this one, and this one is written. There's a couple places it could anneal. It could anneal as written here, or it could be the complement and the flipped over version written this way. Those are our two possible options. Look for is CTTCAA. Up, I think I see that right there. So it looks like it's going to anneal there. And so we should get one fragment that's CTTCAA. Does everybody see that? This piece would get cleaved off. And then for this one's probably going to go the other way, and it should read GCCAGA. And so what that means is I should look this way, and I should look for, seriously? Come on, go back. Okay. I should look for CGG. I'm going to read this way, and I should be looking up here. Haha. CGG. And then I've got, not what I want. Middle, whoa, say that again. Middle, bottom, left. This way? There. Am I there? No. This one. Here. Sorry, guys. CG. Oh, wait. So we have CGG. That does not. Whoa. You guys are. This strand right here. All the way here. Oh, you guys are seeing something I'm not. Where am I going? All the way to the right here. And it goes this. Yes. Oh, wait. Meaning that this primer would need to anneal. It's the same. GCCAGA. There. Okay, okay. Ooh, sorry about that. 1, 2, 3, 4. Good. So that guy as written, and its complement would be this guy. Are we good with that? I don't know if I'm good with that, but are you guys good with that? Yes? All right, on exam day, take your time to write this out. Otherwise, you'll do what I just did, where I was like, I don't know where the heck I'm going. But so this primer 2 is going to anneal on, and we're going to physically flip that over, and it's going to anneal onto this portion. Yes? All right, and then that would go this way, whereas this primer up here would go this way. And so what we'd essentially get is we would get the fragment that's that long. Does that make sense? Or rather, we get the 2 fragments. Does that make sense? Yes? All right, what next? Please not PCR. Yes, 14? Oh, you said number 14? Okay. All right, predict whether creatine kinase will operate in the direction of ATP synthesis or phosphocreatine synthesis at 25 degrees given those concentrations. What is delta G of the reaction under these conditions? Well, we're going to have to plug and chug a little bit here. Oh, is up here. All right, so that's going to be a delta G calculation. We were given a standard one. Is this cleaned off? No. Oh, well. Right. We were given standard state, so we're going to look for non-standard state. And so that's going to be delta G equals 12,600 plus 8.314 times, we're at 25, so 298. And that's going to be the natural log of, and now in this case, now this is a 2 over 2, so you don't have to change the units on this. If it were 2 over 1 or 1 over 2 or 3 over 6 or really anything that's not the same units, you must change to molar. In this case, we don't have to because they'll cancel. But it looks like ADP is going to be on the top, and that's 0.15. Phosphocreatine is 2.5. This is all millimole, right? Cool. ATP is 4, and creatine is 1. And so we divide that and take the natural log and all that junk. And what do we end up getting as a delta G value here? Somebody do the math. Anybody here able to do this in their head? I mean, there are people who can do that. I was just asking. Like, I've had people before who can do like complicated math in their head, and I'm always like really impressed and sort of scared. But we got a number? Yes, no, something. What did you get as a final value? 6,785. So 6.7, and that's positive, yes? Oh, wait. Is everybody good with the math there? If you're not getting this answer on your calculator, that means you need to work with your calculator a little bit, okay? Make sure you can use a calculator. All right. Since that is positive as written, that means that the reaction is not spontaneous in the forward direction. It's spontaneous in the reverse direction. And so the things that are going to be made spontaneously at that concentration are ATP and creatine. That is the spontaneous synthesis. All right? So ATP synthesis is going to be favored, and the delta G value is 6.7. Good? All right. If delta G is negative, then we assume that it's spontaneous as written, and the products are what's going to be made or synthesized. If delta G is positive, then it's going to be the reactants that are formed, and those are what's quote-unquote synthesized. If delta G is 0, the reaction is at equilibrium, and therefore we can say that neither are being synthesized net. Technically, the reactions are still occurring, but the net total is not being changed. Yes, ma'am? Wait, say that one more time. Well, well, ATP hydrolysis, like all on its own, that does have a negative delta G value. But the question is in regards to the given equation. Yeah. So one of the nice things about Dr. Katsuli is that she's not going to expect you to know a bunch of random equations. You should be able to do every question, get any calculation question within the context of what's given. Very rarely does she make you like know random little factoids. Now, if it's a concept or memorization question, you have to know factoids. But for these, she's not going to ask you stuff like this. Dr. K, I cannot say the same. He's a little crazy when it comes to memorizing things, so I'm sorry for that. I remember I struggled through. Yes, ma'am. The equation that's written as is, if delta G is positive, the things that are on the product side, those are synthesized. If delta G is negative, then it's spontaneous that way. If delta G is positive, the reactants are spontaneously synthesized. And so in this case, ATP and creatine are the reactants, so those are synthesized under those conditions. Does that make sense to everybody? Cool. Next question. Well, actually, what time are we at? 5:20 what? Just 5:19? Okay. Yes. Yes. Okay. Calculate the delta G for the redox reaction of cytochrome C and ubiquinol given the standard half reactions below. What is the number of ATP molecules that could theoretically be synthesized under standard conditions? Whew. Okay, so the first thing we want to do is we're given the half reactions as written. Now the first thing we need to note is that what the middle equation there, what are the electrons in regards to the last equation? Are they the same? They're different. We need to double that equation, which means that delta E will also get doubled. Are we all good with that? So our delta E values are going to be 0.4, that's going to be 0.470, and our E2 is going to be 0.045. Good? And it looks like in order to get that final equation, we need to flip the bottom equation. Agree? Which means that this E value will be negative. I'm going to add these together, and so we'll get like 0.435. Is that right? Or is it 25? 25. Positive 0.425. Is everybody good with that? Make sure I did that right. Okay. And that makes sense because it's saying that its ATP is forming, so this should be a positive number. Okay. And then wants to know delta G, and we know this equation. We know delta G standard is equal to negative NF delta E standard. In this case, how many electrons are being transferred? 2. So we're going to say delta G is equal to negative 2 times 96,485 times 0.425, and that gives me a delta G value of what? I'm guessing negative 36,700. Yes, we would divide that by 1,000 to get delta G to equal negative 36.7 kilojoules, because this unit is in joules. All right. Now, that's the amount of energy that's produced. And what the last little bit of information the question is saying is that ATP formation, 30.5 kilojoules of energy are needed to form 1 mole. All right? So in terms of the molecules, we need 30.5 kilojoules. Well, that wouldn't be the same number, but 35 kilojoules to form 1 mole. Does that make sense? So in terms of molecules, we need at least 30.5 intervals to be able to form 1, 2, or 3 molecule. Does that make sense? You could do math with Avogadro's number here, but that's not necessary. All right? We're just going to compare this value with that value. Is this larger than 30.5? Is it twice as large? No. So we can only form one molecule. It needs to be at least twice as large to be able to form 2, 3 times as large to form 3, and so on and so forth. Yes? Yes. Well, I doubled the first one. But the second equation, the delta E standard state, did not. It's written with 2 electrons. So we only double that middle one. Do we get 0.4? Make sure I did all the math right here because I did that in my head. But 0.235 times 2 is 0.47, yes? Oh, that might be a typo. Email her about that. Email and ask her. That may just be a typo. I can attest to once you get past like 3 questions, typos start occurring. It's rather difficult to avoid them. But the way we did it, that should have produced the right answer. We should, well, assuming all this, what did you plug in a 2 here? And you plugged in that number. Huh. Interesting. We're out of time, so I won't be able to work that out. Do we have any last minute quick questions about anything? Yes. Say that again? Well, the video is posted, but that's all I got. Yeah, because no, that's where typos happen and people freak out. Yeah, I've done that before, and I apologize, guys. I don't post the answer keys anymore, like just an answer key, because what happens is I type that, and then I typo, and then someone freaks out and like calls my boss and screams at them over the phone. You think I'm kidding. Someone has done that before. So I don't post answer keys. The video, though, is online. You can go through and watch that. I'm sorry you'll have to listen to another 2 hours of me talking. I know it's like horrible torture, but you will survive. Yes, the multiple choice, those answers are on. All right, good luck for everyone on their final. Dr. Katsuli's class, I will see you guys bright and early Tuesday. Don't forget your finals start at 7 a.m., okay? If you're in Dr. Katsuli's, it starts at 7, and I have no idea what time Dr. Kapashkov's start. Make sure you guys show up on time, though. See you guys. What?